Picture yourself standing in a quiet room, skin exposed to cool air. Beneath that surface silent, invisible, relentless 3 billion heartbeats worth of blood has traveled through vessels that could wrap twice around the earth. Neurons fire in cascades too swift for conscious thought. Electrical storms contained within a skull no thicker than a fingernail. The machinery hums. It breathes. It repairs itself in sleep. But what if this vessel, this improbable collection of bone and synapse, was not merely the product of chance and time? What if, hidden in the of DNA, beneath the languages of adenine and guanine, lies evidence of something else, not divine creation, not pure randomness, but engineering? The human body is a library written in 4 letters, containing instructions older than mountains. 3 billion base pairs spell out eye color, disease resistance, the shape of thought itself. Biologists have mapped this library. They can read its chapters. Yet questions linger in the margins, anomalies that do not fit comfortably into the official story of gradual, unguided change. Consider the brain. 1.5 kilograms of tissue that somehow produces Shakespeare, imagines distant galaxies, weeps at music. It consumes 1/5 of the body's energy, while occupying only 1/50th of its mass. This is no efficient design by nature's usual standards. It is profligate, extravagant, almost deliberate. Evolution, we are told, is a patient sculptor, working over millions of years with no plan, no blueprint, only survival's cruel editing. And yet certain features of human biology suggest not slow accumulation, but sudden leaps, changes that appeared not over epochs, but in evolutionary eye blinks. The skin beneath which you exist, the mind with which you perceive these words, what if they were sketched first, then built? What if humanity's origin story is not only incomplete, but fundamentally misunderstood? This is not a story about ancient aliens or conspiracy. It is something quieter. A meditation on possibilities hidden in the spiral staircase of our genes. A question whispered by the body itself, if only we knew how to listen. The desert stretches before us, vast and patient. The story begins not with answers, but with the oldest human impulse, the need to ask, where did we come from? The accepted narrative unfolds like a map drawn over centuries of excavation and hypothesis. 7 million years ago, somewhere in the forests and savannas of Africa, an ape-like creature made a choice, or rather, a mutation made it for them. They began to walk upright. Bipedalism freed the hands, and the hands eventually shaped tools. This is Sahelanthropus chidensis, Australopithecus afarensis, the long lineage of proto-humans whose bones now rest in museum drawers, labeled and dated. Lucy, the famous skeleton discovered in Ethiopia, is 3 million years old, a child of the Pliocene, walking on 2 legs through a world that knew nothing of cities or agriculture. Natural selection, Darwin's elegant mechanism, is not design. It only eliminates. Those variants slightly better adapted to their environment, faster, smarter, more resistant to disease, survive to pass on their genes. Across generations, populations shift, new traits emerge, old ones fade. Given enough time, a fish becomes a lizard, a shrew becomes a primate, an ape becomes human. The genetic evidence supports this story. Human DNA shares 98.8% similarity with chimpanzees, our closest living relatives. The differences, that slim 1.2%, account for language, abstract thought, and the capacity to wonder about our origins. Mutations, gene duplications, regulatory changes. The tools of evolution, random and unguided, sculpting complexity from simplicity. Fossil records trace the gradual expansion of cranial capacity. Homo habilis, 2.5 million years ago, possessed a brain roughly half the size of modern humans. Homo erectus followed, then Homo heidelbergensis, each iteration slightly larger-brained, slightly more sophisticated. By 300,000 years ago, Homo sapiens emerged in Africa anatomically modern, though not yet behaviorally so. This is the mainstream account, taught in universities, supported by thousands of peer-reviewed studies. It is a story of slow transformation, of incremental adaptation to changing climates and ecological niches. No divine intervention, no guiding hand, only time, chance, and survival. And yet, even within this well-documented narrative, certain details resist easy explanation. Why did brain size triple in only 2 million years? A pace nearly unprecedented in mammalian evolution. Why do humans possess traits that seem almost maladaptive? Difficult childbirth due to large infant skulls? Vulnerability during prolonged childhood? A brain that demands so much energy it becomes a liability during famine? Paleontologists note gaps in the fossil record, missing links that remain missing despite decades of searching. Geneticists discover sequences in human DNA that appear suddenly without clear ancestral precedent. These are not arguments against evolution, but invitations to refine the story. The official narrative stands. But like all maps, it is not the territory itself only our best attempt to chart it. Descend now into the microscopic realm where existence is written. The double helix spirals, elegant and relentless, containing within its structure the accumulated history of every ancestor back to single-celled organisms swimming in primordial seas. But not all passages in this ancient text read smoothly. Human chromosome 2 is an anomaly. All great apes, chimpanzees, gorillas, orangutans possess 24 pairs of chromosomes. Humans have only 23. This is not a trivial distinction. Chromosomal number is typically stable across species. Changes are rare and often catastrophic. And yet, at some point in our lineage, two ancestral chromosomes fused together, merging into a single larger structure. Geneticists can identify the fusion site, the telltale traces of genetic glue where the two chromosomes joined. This event likely occurred between 5 and 6 million years ago, after the split from our common ancestor with chimpanzees. It was, by all accounts, extraordinarily unlikely, and yet it happened. And somehow, the organism carrying this mutation survived to reproduce. Mainstream science explains this as a rare but natural occurrence, a chromosomal accident that did not harm reproductive fitness and thus persisted. But the specificity of it, the precise fusion, the survival against odds, leaves a faint, unanswered question hanging in the genetic literature. Elsewhere in the genome, other peculiarities emerge. The FOXP2 gene, crucial for speech and language, underwent rapid mutations in the human lineage within the last 200,000 years. Two specific amino acid substitutions differentiate human FOXP2 from the chimpanzee version, and these changes are associated with fine motor control of the larynx and face, the very muscles required for articulate speech. Evolution does not usually move this fast. The average mammalian gene evolves at a steady, predictable rate. But FOXP2 and several other human-specific genes show evidence of positive selection, meaning they were advantageous and spread rapidly through populations. Why? What environmental pressure suddenly rewarded complex communication so intensely that mutations conferring it were preserved and amplified? Then there is the brain itself. Between Homo habilis and Homo sapiens, cranial capacity nearly tripled in less than 2 million years. In evolutionary terms, this is a sprint. Brain tissue is metabolically expensive. It requires abundant nutrition and carries risks, difficult childbirth, extended childhood dependency. For such a costly organ to expand so rapidly suggests intense selective pressure, or more mysteriously, some other factor accelerating change. Junk DNA, the stretches of the genome once thought to serve no function, has been re-examined in recent decades. It turns out much of it is regulatory, controlling when and how genes are expressed. Some researchers have noted patterns within these regions that do not appear in other primates, structures that emerge suddenly in the human lineage. Could these be scars of ancient editing? Echoes of intentional modification buried so deep in time that their origins are lost? The genome is not a smooth narrative. It is punctuated, interrupted, filled with chapters that begin abruptly and without clear preamble. Mainstream science attributes these to natural processes we do not yet fully understand. But understanding, by definition, is incomplete. Meanwhile, the genes continue their silent testimony, waiting for the right questions. There are places in the story where the light goes dark. Gaps in the fossil record, yes, but also deeper silences. Stretches of time where humanity existed, and yet left almost no trace of itself. For hundreds of thousands of years, Homo sapiens wandered the African continent, anatomically modern but mysteriously quiet. Archaeology tells us that modern humans emerged roughly 300,000 years ago. But for 250,000 of those years, they created little more than simple stone tools, maintained small nomadic groups, and left behind scant evidence of symbolic thought or culture. Then, approximately 50,000 years ago, something changed. This period, often called the Great Leap Forward or the Upper Paleolithic revolution, marks an explosion of human creativity. Cave paintings appear in France and Spain, intricate and expressive. Carved figurines emerge from archaeological sites across Europe. Tools become specialized and sophisticated. Jewelry, ritual burial, evidence of music and storytelling, all within a span of a few thousand years. What ignited this transformation? Biologically, humans were no different than their ancestors 200,000 years earlier. The brain had not grown larger, the vocal apparatus had not changed, and yet behavior shifted radically and rapidly, as though a switch had been flipped. Some researchers propose that a genetic mutation enhanced cognitive fluidity, allowing humans to combine different types of knowledge in novel ways. Others suggest that population growth led to increased cultural transmission, a feedback loop of innovation and learning. Still others wonder if climate change, the harsh conditions of the last ice age, forced humanity to adapt or perish. But none of these explanations fully account for the speed and breadth of change. Agriculture, for instance, appeared independently in multiple locations around the world: Mesopotamia, China, Mesoamerica roughly 10,000 to 12,000 years ago. For 3 million years, hominids survived as hunter-gatherers. Then, almost simultaneously across continents, disparate groups began cultivating grains, domesticating animals, building permanent settlements. Coincidence? Parallel evolution of cultural complexity? Or something else? Ancient Sumerian texts speak of the Anunnaki, beings who descended from the heavens and taught humanity agriculture, writing, and law. Egyptian mythology describes gods who walked among men, imparting knowledge of medicine and astronomy. Hindu scriptures recount the Devas, celestial architects who shaped civilization. These myths span cultures that had no contact with one another, and yet the themes are strikingly similar. Advanced beings descending from the sky, gifting humanity with knowledge. Modern scholars dismiss these stories as allegory, the primitive mind's attempt to explain natural phenomena. But myths are also memory distorted, mythologized, but sometimes rooted in real events. What if these stories preserve an echo of something that happened? Not divine gods, but visitors or engineers who intervened at a crucial moment? The silence of our past is not empty. It is filled with whispers we have not yet learned to hear. Pause here in this brief interlude and consider an odd fact that neuroscientists have long pondered. The human brain, despite accounting for only 2% of body mass, consumes 20% of the body's total energy. This is an extravagant allocation, unprecedented among mammals. A resting brain burns roughly 320 calories per day more than a quarter of an average adult as basal metabolic rate. During intense cognitive work, this demand increases even further. From an evolutionary standpoint, this is a dangerous gamble. In environments where food is scarce, such a hungry organ becomes a liability. A creature whose survival depends on constant foraging cannot afford to feed a brain that offers no immediate survival advantage. And yet, humanity's brain expanded rapidly at great metabolic cost. Compare this to other intelligent species. Dolphins, whose cognitive abilities rival ours in some domains, have brains that consume roughly 8 to 10% of their energy budget high, but not extreme. Elephants, with their massive brains, allocate even less proportionally. Humans are outliers, and outliers in evolution usually signal either intense selective pressure or evolutionary dead ends. We did not become a dead end. We thrived. But the question remains, what advantage did this energy-hungry brain provide that justified the cost? Language, problem-solving, social cooperation, all plausible answers. But these traits could have been achieved with a more energy-efficient organ. The human brain is not merely functional. It is over-engineered, capable of abstract mathematics, symphonic composition, and existential philosophy, none of which directly enhance survival in the ancestral environment. Unless, of course, survival was not the only goal. If one were designing a species for a specific purpose, say, technological advancement or cultural development, an oversized, energy-intensive brain would make sense. It would be a feature, not a bug. A deliberate investment in cognitive capacity, even at the cost of short-term survival efficiency. This is speculation, of course. Mainstream science explains the brain's expansion through sexual selection, social competition, and niche construction. But the paradox remains. Why is the human brain so powerful, so costly, and so seemingly unnecessary for basic survival? The neurons fire, consuming their glucose, and the question lingers like a shadow. Imagine for a moment that you are an engineer tasked with designing a species, not creating life from nothing that remains beyond reach, but modifying an existing template, an upright ape with opposable thumbs and social tendencies. What traits would you prioritize? First, intelligence. Not merely problem-solving ability, but abstract reasoning, the capacity to imagine what does not exist and then bring it into being. You would enhance the prefrontal cortex, expand working memory, fine-tune neuroplasticity to allow rapid learning and adaptation. Second, communication. A species capable of complex thought must be able to share that thought with others. You would modify the vocal apparatus, introduce genetic changes like those seen in FOXP2, and wire the brain for grammar, syntax, recursion, the architecture of language. Third, cooperation. Intelligence and communication are useless if individuals cannot work together toward common goals. You would amplify empathy circuits, strengthen mirror neurons, and introduce mechanisms for social bonding oxytocin receptors, facial recognition, the capacity for shared intentionality. Fourth, adaptability. The species must survive in diverse environments, deserts, tundras, rainforests. You would preserve genetic diversity, maintain a flexible metabolism, and ensure the organism can modify its surroundings through tool use and technology rather than waiting for biological evolution. Fifth, longevity and health. A long lifespan allows accumulated knowledge to be passed down, traditions to form, civilizations to stabilize. You would introduce DNA repair mechanisms, robust immune responses, and perhaps even regulatory genes that delay aging. Now look at humanity. Look closely. We possess all of these traits. Not perfectly, biology is messy, full of trade-offs and legacy structures, but we possess them in configurations that seem almost purposeful. The opposable thumb, binocular vision, upright posture, these are inherited from primate ancestors. But layered atop this foundation are enhancements that push us far beyond any ecological niche we originally occupied. The human hand is not merely a grasping tool. It is capable of micromanipulations, threading a needle, performing surgery, painting the Sistine Chapel ceiling. The human eye does not just see. It interprets art, reads written language, detects minute facial expressions that convey emotional states. Our genome contains roughly 20,000 protein-coding genes, but the complexity emerges not from the number of genes, but from their regulation, how they are turned on and off, combined and recombined. Humans share vast amounts of genetic material with other species, but small regulatory changes create profound differences. If someone did engineer us, they would not need to build from scratch. They would only need to edit, insert a regulatory sequence here, duplicate a gene there, fuse 2 chromosomes, accelerate a few key mutations. The result would look exactly like what we see when we study the human genome. A mosaic of ancient and recent, familiar and strange, structured yet filled with inexplicable quirks. A blueprint, perhaps, or the remnant of one. The gene they examine is called FOXP2, a sequence of roughly 2,200 base pairs that codes for a transcription factor, a protein that regulates other genes. This gene exists in many animals: mice, songbirds, bats, primates. But in humans, it is different. 2 amino acid substitutions, just 2, out of the 715 amino acids that make up the FOXP2 protein separate humans from chimpanzees. Individuals born with mutations in FOXP2 suffer from severe language impairments. They struggle with grammar, articulation, and sequencing sounds into coherent words. The gene is essential for spoken language, but it also affects other motor skills, facial expressions, hand gestures, even written communication. Researchers introduced the human version of FOXP2 into mice. The mice developed altered vocalizations, more complex patterns of squeaks, and ultrasonic calls. Their brain structures changed subtly, particularly in regions associated with motor learning. The gene, it seems, does not create language outright, but it lays the neural groundwork for it. The mutations in FOXP2 were positively selected, meaning they spread rapidly through human populations because they provided a survival advantage. A single nucleotide altered here, another there. The protein folds slightly differently, binds to regulatory regions with altered affinity, and suddenly the brain can coordinate breath, tongue, and larynx in ways no other species can. Consciousness remains neuroscience's deepest mystery. How does electrochemical activity in the brain produce the sensation of being? The taste of coffee, the ache of loneliness, the sudden arrival of an idea, none of these experiences should emerge from neurons firing across synapses. Consider the structure of human emotion. We do not merely feel hunger or fear, drives shared by all animals. We experience guilt, awe, existential dread, aesthetic rapture. These emotions are costly, often maladaptive. Guilt inhibits selfish behavior, but also paralyzes decision-making. Awe inspires reverence, but leaves individuals vulnerable, distracted by beauty or grandeur. Why would evolution, that ruthless editor, preserve such traits? Why? Empathy is useful for cooperation, yes, but it is also a vulnerability. No other species does this. Bowerbirds build elaborate structures to attract mates, but they do not paint landscapes or compose symphonies. Human creativity often serves no immediate function. It is expression for its own sake, the mind playing with possibility. Dreams, too, are peculiar. Other animals dream, but human dreams are narrative, symbolic, emotionally rich. We wake from them disoriented, having lived entire lives in the span of minutes. Some neuroscientists propose that dreaming consolidates memory or processes emotion. But the content of dreams often defies logic, offering no clear adaptive benefit, unless once more we consider design. If an engineer wished to create a species capable of rapid cultural evolution, they would need to instill certain traits: empathy for social cohesion, creativity for innovation, consciousness for self-reflection, and moral reasoning. These traits would not be perfect. Evolution never produces perfection, but they would be exaggerated, pushed beyond the baseline seen in other species. The human mind, in this light, is not merely an organ for survival. It is an instrument for something else: exploration, creation, understanding. It is a tool that has become aware of itself, and that awareness is asking, who made this tool and why? The question reverberates through the corridors of thought, unanswered. Within its 3 billion base pairs lie fragments of ancient viruses, duplicated genes, regulatory sequences whose purpose remains obscure. Some regions are scaffolding, structural elements that maintain chromosomal integrity. Others remain mysterious. Endogenous retroviruses, ERVs, are one such mystery. These are viral sequences that infected our ancestors millions of years ago, inserting themselves into the genome. Normally, such insertions would be harmful, but occasionally they become beneficial, co-opted by evolution for new purposes. But some sequences defy easy categorization. There are stretches of DNA with unusually low mutation rates, suggesting they are under selective pressure to remain unchanged. There are palindromic sequences, genetic phrases that read the same forward and backward with no known function. There are patterns that some researchers claim resemble mathematical structures, though mainstream science remains skeptical. In 2013, a team of geneticists published a provocative paper suggesting that certain non-coding regions might contain information-rich structures, possibly remnants of horizontal gene transfer genes acquired not from ancestors, but from other organisms. Horizontal gene transfer is common in bacteria, but rare in complex organisms. Yet the human genome contains several sequences that appear to have no primate precedent. Where did they come from? Mainstream explanations invoke ancient viral infections, chromosomal duplications, or undetected common ancestors. But the door remains slightly ajar for more speculative ideas. If one were to leave a signature in a genome, a marker indicating intentional design, how would it be done? An obvious message would degrade over time, erased by mutation and genetic drift. But a subtle pattern, embedded in regulatory regions or disguised as junk DNA, might persist for millennia. And yet, the genome is full of whisper sequences that do not quite fit, patterns that seem too orderly to be random, and functions that remain stubbornly unclear despite decades of research. The double helix continues its spiral, indifferent to interpretation. Whether the work of blind evolution or distant architects, it carries the story forward, one generation at a time. There are genetic sequences in the human genome that, by all conventional measures, should not be there. They appear suddenly in our lineage, with no clear ancestral precedent in earlier hominids. These are not errors or artifacts of sequencing. They are real, functional, and inexplicable. One example, the HAR1 gene, or Human Accelerated Region 1. This sequence is 118 base pairs long and is located on chromosome 20. It is highly conserved across vertebrates, meaning it has remained almost unchanged for hundreds of millions of years in species from chickens to chimpanzees. But in humans, it underwent rapid mutation, accumulating 18 changes in just 6 million years. HAR1 is expressed during fetal brain development, particularly in the cortex. The changes in this region are associated with the expansion and folding of the human brain, the very feature that distinguishes us most from other primates. The standard answer: positive selection. The mutations were advantageous, allowing for greater cognitive capacity and thus spread through the population. But the speed and specificity of these changes remain striking. Then there is RGAP11B, a gene unique to humans and absent in other apes. It arose from a partial duplication of an existing gene, RGAP11A, roughly 5 million years ago. But the duplicated version acquired new functions. It promotes the proliferation of neural stem cells, leading to a larger neocortex. A single gene, appearing suddenly, capable of reshaping brain structure. How did such a gene arise through random mutation? How did it avoid being silenced or degraded, as most gene duplications are? And then there are the endogenous retrovirus's ancient viral DNA integrated into our genome. Some of these sequences are active, producing proteins essential for placental development. Without them, human pregnancy would not be possible. But not all ERVs are accounted for. Some appear in human DNA, but not in closely related species, suggesting they were acquired recently or introduced. Mainstream genetics explains these anomalies through mechanisms like horizontal gene transfer, gene duplication, and regulatory evolution. These are valid, well-supported explanations, but they also leave room for wonder. If someone wished to modify a species subtly, they might introduce small, functional sequences at key points in development genes that enhance brain growth, improve immune response, or enable new capacities. Over time, these sequences would blend into the genome, indistinguishable from naturally occurring mutations. The footprints are there, faint but persistent. Whether they lead back to the slow march of evolution or to something else, no one can say with certainty. And yet, the timing nags at the edges of explanation. But bottlenecks are also opportunities. Small populations experience rapid genetic change because beneficial mutations spread quickly. If a few individuals carry advantageous traits, intelligence, adaptability, social cohesion, those traits can dominate the gene pool within a few generations. Could a bottleneck have been engineered? Not naturally, but intentionally, a population reduced to a small, carefully selected group, then guided toward specific traits? This veers into deep speculation, the territory where evidence thins and imagination fills the gaps. But the genetic data is clear. At some point, humanity passed through a narrow gate, and only a handful of lineages emerged on the other side. Mitochondrial Eve did not know she would become the mother of all humanity. She lived, reproduced, and died, unaware that her mitochondrial DNA would carry forward through millennia while countless others faded into genetic oblivion. Was she special, or simply lucky? The mitochondria in your cells, inherited from your mother, who inherited them from hers, stretching back through an unbroken chain to that one woman in Africa, offer no answer. They simply continue their work, powering the cells, burning glucose, sustaining the lineage. In ancient Sumer, carved into clay tablets 5,000 years old, scribes recorded the story of the Anunnaki beings who descended from the heavens to earth, bringing knowledge of agriculture, mathematics, and law. The tablets describe how these beings created humans to serve as workers, mixing the clay of the gods with the essence of the gods. In Egypt, hieroglyphs tell of Thoth, the ibis-headed god of wisdom, who taught humanity writing, medicine, and astronomy. The texts are explicit. Thoth descended from the sky, walked among humans, and imparted knowledge that transformed civilization. In the Rig Veda, one of Hinduism's oldest texts, the Devas are celestial beings who shaped the world and guided human development. They are not omnipotent gods, but advanced entities with specific roles and limitations. The texts describe flying chariots, powerful weapons, and detailed cosmological knowledge. In the Americas, the Popol Vuh of the Maya recounts how the gods created humans after several failed attempts, each iteration more refined than the last. The final creation required mixing maize with the blood of the gods, producing beings capable of thought and speech. Scholars classify these stories as mythology cultural narratives that explain origins, reinforce social norms, and encode moral lessons. And yet, the similarities across these geographically isolated cultures are striking. Sky beings. They are specific, detailed, and recurring. Could these myths be distorted memories? Not of gods, but of something real visitors, perhaps, or an earlier advanced human civilization that collapsed, leaving only fragmented oral traditions? The mainstream dismisses such ideas as pseudoscience, the domain of conspiracy theorists and ancient astronaut enthusiasts. And much of that criticism is valid. There is no hard evidence for ancient extraterrestrial contact, no artifacts that cannot be explained by human ingenuity. But myths are persistent. They survive millennia, passed down through oral tradition, and eventually codified in writing. They shape cultures, justify rulers, and explain the inexplicable. And sometimes rarely, but sometimes they preserve kernels of truth, events distorted beyond recognition, but not entirely invented. The flood myth, for instance, appears in cultures worldwide. Geology confirms that catastrophic floods did occur at the end of the last ice age as glaciers melted and sea levels rose. The specific details, Noah's Ark, Utnapishtim's boat, Manu's fish, are mythological elaborations, but the core event may have been real. But something that seemed godlike to ancient people's advanced technology, perhaps, or knowledge so profound it appeared divine. The tablets, scrolls, and steles offer no definitive answer. The Dogon people of Mali, for instance, have long spoken of Sirius B, a white dwarf star invisible to the naked eye, discovered by modern astronomers only in the 19th century with the aid of telescopes. The Dogon claim this knowledge was given to them by amphibious beings called the Namo, who came from the Sirius system. Skeptics note that Western anthropologists visited the Dogon in the early 20th century and may have inadvertently shared astronomical knowledge, which was then incorporated into local mythology. This is plausible, but the Dogon insist their knowledge is ancient, predating European contact. In South America, the Nazca lines stretch across the Peruvian desert enormous geoglyphs depicting animals, geometric patterns, and straight lines that run for miles. Why create art that cannot be seen from the ground? Some researchers propose the lines were ritual pathways or astronomical calendars. Others suggest they were offerings to sky gods, meant to be seen from above. These are old questions, asked by mavericks and outsiders, dismissed by mainstream academia. And yet, they refuse to die. The Earth is no stranger to cataclysm. Its history is punctuated by mass extinctions, climate upheavals, and sudden environmental shifts that reshaped life's trajectory. The end of the last ice age, roughly 12,900 years ago, marked one such upheaval, the Younger Dryas. The cause remains debated. Some scientists propose that massive meltwater from retreating glaciers disrupted ocean currents, particularly the Atlantic meridional overturning circulation, plunging the northern hemisphere into cold. Human populations had been decimated, but those who survived emerged into a transformed landscape, and within a few millennia, agriculture appeared. Could catastrophe have been a crucible? A selective pressure so intense that only the most adaptable, intelligent, and cooperative humans survived? Early agriculture was harder than hunting and gathering, more labor-intensive, less nutritious, more prone to famine. Why did humans adopt it? This idea appears in ancient myths, the Sumerian texts describing the mixing of divine essence with clay, the Greek legends of Prometheus shaping humans from earth and fire, the Hopi stories of the sky people who mingled with Earth's inhabitants. Modern proponents of this theory point to anomalies in human physiology. but it persists, whispering in the margins of biology and anthropology. What if humanity is a hybrid part terrestrial ape, part something else? What if the anomalies in our genome, the sudden leaps in intelligence and behavior, the myths of celestial teachers, what if they all point to the same thing? Contact? Modification? Engineering? The body offers no clear answer. It simply continues, breathing, pumping blood, firing neurons, oblivious to the questions swirling around its origin. In a small village in northern India, a boy named Raj begins speaking at age 3. His first words are not in Hindi, his family's language, but in fluent Bengali, which no one in the household speaks. He describes a house by a river, a woman with a scar on her left hand, a dog named Balu. His parents, bewildered, investigate. They travel to a village 200 miles away, following the child's descriptions. There, they find the house, the river, the woman with the scar. She is the widow of a man who died 5 years earlier, 2 years before Raj was born. The dog, Balu, had died with him. The woman weeps, recognizing details only her late husband could have known. Raj describes the interior of the house, the location of a hidden cash box, the name of a childhood friend. All correct. Cases like Raj's are documented in the literature of reincarnation studies, a fringe field dismissed by mainstream science, but meticulously catalogued by researchers, like Dr. Ian Stevenson of the University of Virginia. Stevenson collected over 3,000 cases of children who claimed to remember previous lives, many with verifiable details. Skeptics propose alternative explanations: cryptomnesia, unconscious memory of forgotten information, coincidence, parental coaching, or fraud. These explanations account for some cases, but not all. Another possibility, rarely discussed: genetic memory. Could certain experiences, emotions, or knowledge be encoded not in conscious memory, but in DNA, passed from parent to child in ways we do not yet understand? Epigenetics, the study of how genes are turned on or off by environmental factors, has revealed that experiences can alter gene expression, and some of these changes can be inherited. Trauma, diet, stress all leave marks on the genome that affect offspring. Could memory itself, in some form, be heritable? Not the full content of consciousness, but fragments, impressions, skills, knowledge woven into the genetic code? Or perhaps the explanation is simpler and stranger. Consciousness is not confined to the brain. It is a field, a pattern, a wave that can, under rare circumstances, persist and re-emerge in a new biological vessel. This is metaphysics, not science, but the cases remain. The children speak, remember, know things they should not know. And we, limited by our current understanding, can only listen and wonder. Raj, now grown, no longer speaks of the river house or the woman with the scar. The memories, if they were memories, have faded. He lives an ordinary life, unremarkable except for a few years in childhood when he seemed to be two people at once. The genome, silent, offers no comment. Assume for a moment that the speculation is true. Assume that humanity was intentionally modified, genetically engineered at some point in our distant past. The immediate question is, why? Not how we have explored possible mechanisms, however speculatively. But why would anyone do this? What purpose would drive such an undertaking? One possibility: curiosity. An advanced civilization, encountering a promising primate species, might experiment not maliciously, but scientifically. They would edit, observe, iterate, seeking to understand the principles of intelligence, consciousness, and culture. Another possibility: utility. Perhaps humanity was designed for a specific purpose: labor, companionship, exploration. The Sumerian texts suggest this: humans created to work the fields, freeing the gods from toil. A grim thought, but not without precedent. Humans have engineered countless species for specific purposes, dogs for hunting, cattle for food, horses for transport. A third possibility: preservation. Perhaps an ancient civilization facing extinction sought to preserve their knowledge, their essence, by encoding it into a durable species one that could survive, adapt, and eventually rediscover what was lost. Or perhaps the motive was altruistic. A spacefaring species, encountering a world rich with life but lacking sentience, might choose to uplift a promising lineage, gifting intelligence and self-awareness not for their benefit, but for ours. There is also the possibility of accident. Genetic modification, even by advanced beings, is risky. What was intended as a minor adjustment might have cascaded into unforeseen changes, creating intelligence as a side effect rather than a goal. Finally, there is the darkest possibility: experimentation without consent, without care for the consequences. Humanity as a project, an experiment, observed from a distance, but not guided, allowed to flourish or fail on our own. These are scenarios drawn from imagination, not evidence. But if we entertain the hypothesis of design, motive becomes central. Intentions shape outcomes. An experiment is different from a gift, and a gift is different from a tool. And if we were designed, does it matter? Does knowing the purpose change who we are, what we have become? Humanity has spent millennia believing we were created by gods, only to discard that belief in favor of evolution. Now some speculate we were engineered, a middle ground between divine creation and blind chance. But in the end, the origin matters less than the outcome. We are here. We think, feel, create, destroy, love, and wonder. Whether by nature, by design, or by some combination, we have become something unique as species, capable of reflecting on its own existence and asking, why? The answer, if it exists, is not written in the stars or the genes. It is written in what we do with the gift or accident of consciousness. We stand now at a threshold. For the first time in history, humanity possesses the tools to edit its own genome deliberately, precisely, and with intent. CRISPR-Cas9, discovered in 2012, is a molecular tool adapted from bacterial immune systems. It allows scientists to cut DNA at specific locations, remove undesirable sequences, and insert new ones. The implications are staggering. Genetic diseases that have plagued families for generations: sickle cell anemia, cystic fibrosis, Huntington's disease, can now be corrected at the embryonic stage. Crops can be engineered for drought resistance, higher yield, and improved nutrition. Mosquitoes can be modified to resist malaria, potentially saving millions of lives. But the technology also opens darker doors. In 2018, a Chinese scientist named He Jiankui announced he had edited the genes of twin girls, making them resistant to HIV. The announcement sparked global outrage. The edits were reckless, unnecessary, and violated international ethical guidelines. He was sentenced to 3 years in prison. But the genie is out of the bottle. The technology exists, and it will not be uninvented. Within decades, possibly sooner, humanity will face the question: should we engineer ourselves? And if so, how? Enhancement, not just correction. Intelligence, strength, longevity, appearance, all could be modified. Designer babies, once the realm of science fiction, are now technically feasible. But who decides what constitutes an enhancement? Who regulates access, ensuring that genetic modification does not become the privilege of the wealthy, creating a biological aristocracy? And what unintended consequences might arise from editing the genome? A system of staggering complexity that we only partially understand. Evolution has shaped humanity over millions of years, a slow, blind process that nevertheless produced resilience, diversity, and adaptability. Genetic engineering, by contrast, is fast, deliberate, and narrow. We risk optimizing for traits we value today while discarding genetic diversity that might be crucial for future survival. And there is an irony here, a closing of the loop. If humanity was engineered in the distant past, we are now becoming the engineers. We are retracing steps someone else may have taken long ago, wielding the same tools, facing the same ethical dilemmas. Perhaps this is inevitable. Perhaps intelligent species, given enough time, always reach this point, the ability to direct their own evolution, to take control of their biology. Or perhaps we are completing a cycle, fulfilling a purpose encoded in our genes from the beginning, to become the designers of life itself. The CRISPR labs hum quietly, the molecular scissors snipping and splicing, rewriting the code. The future is being edited, one base pair at a time. Imagine a world, not distant, where genetic modification is commonplace. Parents select traits for their children, not just the absence of disease, but enhanced memory, improved athletic ability, even temperament adjustments. The technology, initially expensive, gradually becomes affordable, integrated into standard prenatal care. Within a few generations, the human species begins to diverge. Those with access to enhancement pull ahead, their children smarter, healthier, longer lived. Those without fall behind, creating not just economic inequality, but biological inequality, a gap that cannot be bridged through education or policy. This is one possible future, dystopian and troubling. But there are others. Perhaps humanity adopts international regulations, ensuring that genetic enhancements are available to all, not just the wealthy. Genetic modification becomes a public good, like vaccination, aimed at improving the baseline health and capability of the entire species. Or perhaps we choose restraint. Recognizing the risks, humanity collectively decides to limit genetic engineering to disease prevention, avoiding enhancement and preserving natural genetic diversity. This would require unprecedented global cooperation, but it is not impossible. Another scenario, transhumanism. Humanity does not stop at genetic modification, but integrates technology directly into biology neural implants, synthetic organs, brain-computer interfaces. The line between human and machine blurs and eventually dissolves. We become something new, post-human, transcending our biological origins entirely. Evolution, for all its blindness, is patient and flexible. It does not optimize for a single goal, but for adaptability, for the capacity to survive unexpected changes. Genetic engineering, by contrast, is narrow, focused on specific traits. Evolution hedges its bets through diversity. Engineering, by its nature, reduces diversity in favor of optimization. We must be cautious, deliberate, and humble. The age of self-directed evolution is upon us. The choices we make in the coming decades will shape not just our civilization, but our species. We will decide what humanity becomes, and we will live or fail to live with the consequences. The spiral of DNA, once the work of nature or distant architects, now sits in human hands. We are the engineers. The question is, will we be wise enough to wield the tools? But if we were engineered, if someone long ago modified our genome, would they not leave a signature? A mark, subtle but intentional, indicating that this was not accident but design? The double helix spirals, silent and patient. It has carried humanity from the African savannas to the moon, from caves to cities. Whether it also carries a message is a question we may one day answer. Until then, it remains a bottle adrift in the sea of time, its contents unknown. The body remembers what the mind forgets. It carries within its structure the history of every breath, every wound, every adaptation its ancestors made to survive. Bones thicken in response to stress. Muscles grow or atrophy, depending on use. The immune system learns from each infection, building a library of defenses. But deeper still, beneath the level of individual experience, the body carries evolutionary memory. The vestigial tailbone, remnant of a tail our distant ancestors once possessed. The erector pili muscles that raise goosebumps, useless now, but once capable of fluffing fur for warmth or intimidation. We walk through life largely unconscious of this heritage. The heart beats, the lungs expand, neurons fire. We take it for granted, this improbable machinery that sustains consciousness. But occasionally, in moments of illness or injury, we are reminded of the body's fragility and complexity. A broken bone mends, cells dividing and calcifying according to instructions written millions of years ago. A wound closes, inflammation giving way to new tissue, guided by molecular signals we are only beginning to understand. The body is both ancient and new, a vessel carrying the accumulated wisdom of countless generations. Whether that wisdom was guided solely by nature or also by intention, it is a legacy we inherit simply by being alive. And in the end, perhaps the question of origin matters less than the question of stewardship. We are, each of us, temporary inhabitants of this vessel. What we do with it, how we care for it, the choices we make about its future, these are the questions that define us. The body speaks, if we listen, not in words, but in the quiet language of blood and bone, breath and heartbeat. Night falls over the desert, the sky vast and dark, pierced by stars that have watched the rise and fall of countless species. Somewhere in that expanse, light left its source eons ago, traveling through the void to arrive here, now, to be seen by human eyes, capable of recognizing it, measuring it, understanding what it means. This capacity to look at a point of light and comprehend distance, time, physics, is what sets humanity apart. Not strength, not speed, but the ability to ask questions and, occasionally, to find answers. The question posed at the beginning remains: what if humans were genetically engineered? We have explored possibilities, examined evidence, wandered through speculation and science, myth and modernity. The answer, if there is one, is not simple. Evolution, as currently understood, accounts for human origins. The fossil record, genetic analysis, comparative biology all support a gradual natural process. Mutations, selection, drift, time. These are sufficient to explain the rise of Homo sapiens from primate ancestors. And yet, anomalies persist. Sudden leaps, unexplained sequences, myths that whisper of sky beings and imparted knowledge. These do not disprove evolution, but they invite deeper inquiry. If we were engineered, it was subtle, woven into the fabric of natural processes so seamlessly that the seams are nearly invisible. If there was intervention, it was gentle, more nudge than creation, more guidance than command. Or perhaps we are alone, always have been, and the anomalies are merely the rough edges of a story we do not yet fully understand. Evolution is complex, full of contingencies and accidents that retrospectively seem purposeful. The human brain evolved not because it was destined to, but because a series of environmental pressures and genetic accidents aligned in just the right way. Both narratives are, in their way, awe-inspiring. To be the product of blind chance, the universe's accidental self-reflection, is humbling and profound. To be the result of intentional design, a project undertaken by beings we cannot imagine, is equally humbling, equally profound. Perhaps the truth lies somewhere in between, or beyond the binary of chance and design. Perhaps the universe itself is structured in such a way that intelligence, consciousness, and curiosity inevitably emerge, given sufficient time and complexity. We stand now at the threshold of becoming engineers ourselves. The tools are in our hands. The genome is an open book, and we are learning to edit it. What will we write? Will we repeat the mistakes of natural evolution, creating fragility in the pursuit of short-term advantage? Or will we proceed with caution, humility, and wisdom, recognizing that we are shaping not just individuals, but the future of our species? The stars offer no guidance. They shine indifferent and ancient, as they always have. But humanity is no longer bound to wonder in ignorance. We have tools, science, reason, imagination, and the capacity to chart our own course. Whether we were engineered or evolved, we are now the authors of our own story. The next chapter is ours to write, and so the question does not demand an answer. It demands reflection, curiosity, and the courage to keep asking. If you've stayed with this journey to the end, if these questions have stirred something within you, consider joining others who wonder about our origins and future. Subscribe, and let's continue exploring the mysteries together. The desert is silent. The stars turn. The story continues.